rest. Okay, so let's go back. And I showed you that picture, and then I say operon on. This operon is on when the repressor is not active, does not attach to operator. The yellow one is called the operator. And then, of course, the RNA polymerase can go through and make, this is DNA, right? The gene for tryptophan E, tryptophan D, tryptophan C, B, E, D, C, B, E. These are protein molecules. This is the mRNA transcription. And then, of course, the RNA polymerase is responsible for it, making it up. Voila, we are done. However, if there is ample tryptophan, that tryptophan attaches to the repressor, and then repressor will go and attach to the operator. It was not labeled, to the operator, and then RNA polymerase cannot go through. Am I making some sense a little bit? It is blocked. So the gene is off. Operon, the genes, all of the genes are off. The operon, the whole thing, the operon, the operon, it is the operator, and then we'll talk about a few of the genes. A few other things are inside of the operator as well, inside of the operon too. Okay, so let's go back and talk about these two things, repressible and inducible operons. So two types of negative gene regulations. Repressible operon is one that is usually on. Repressible, they're usually on. Binding of a repressor to the operator shuts off transcription. The TRP, tryptophan, of course, you will see a few other, or one of these. That's a tryptophan operon, is a repressible operon. An inducible operon is one that is usually off. So inducible is off, and repressible is usually on. A molecule that is called inducer inactivates the repressor and thus on the transcription. The LAC, L-A-C, is lactose. You know, TRP is tryptophan, LAC. They use this a lot in biology when you're going to go do research, and you will see these terms, LAC, TRP. They usually take the three letters, and that's lac operon. It means lactose. And what was lactose? From the beginning of semester, it was what? It's a disaccharide. Remember that? It is found in milk. Lactose is a disaccharide that is found in the milk. So operon is an inducible operon and contains genes that code for enzymes used in the hydrolysis and metabolism of lactose. By itself, the lactose repressor is active and switches the lac operon off. A molecule called an inducer inactivates the repressor to turn the lac operon on. Here we go. So another example of negative control, you have this lac. Is that right? No, AIC, a regulatory gene. That's a regulatory gene. That's a promoter right here. And this is your inactive repressor right here. And this is your RNA polymerase and LAC-Z. We have LAC-Z, LAC-Y, and LAC-A. And I will talk about, you will see this. I wish, I'd like to give you a big picture, and then we'll talk about details of it here. So this operon is off because that inactive repressor attaches to the operator. So the RNA polymerase cannot go through. That's another example of a negative control. The other one, the operon is on, and I'll show you a big picture of them. So they call it allolactose. Allolactose, remember in first exam, you studied the glycosylic bond between the sugar molecule and sugar molecule. There is a covalent bond called 1-4. In case of alpha, is glycosylic bond. You remember that? In this case, the lactose is, they call it allolactose. Allo, it means different than lactose, but it is actually lactose is made up of sugar, it's made up of glucose and galactose. You remember that? Lactose is made up of glucose and galactose. This one is 1 to 6 alpha linkage. The real lactose in the milk you drink is 1 to 4 alpha linkage. This one is 1 to 6. But anyhow, don't worry about that. So allolactose attaches to the repressor, and then what happens? Then it makes it inactive. When this repressor is inactive, then RNA polymerase start moving along, going downstream, and making up these protein molecules. Okay? Eventually, RNA is synthesized, and the mRNA synthesized, and mRNA make up this molecule. Let's go ahead and look at it one by one. So you have a regulatory gene right here. You have the promoter from here to here, and that would be your operator right here. So when this active repressor, a protein molecule, of course, all of these repressors and inducers are protein molecules. When this protein molecule attaches the operator on DNA molecule, this is DNA, remember? This is DNA right here, all of that is DNA. And when it attaches that segment of DNA, which is called operator, then RNA polymerase is inactive. Does not do transcription. There is no transcription. So we call this operon off. This operon from here to here is off. Now, how does the same genes, same thing, LAC-Z, you see LAC-Z, lactose Z, and after that they didn't draw it in here, LAC-Y and LAC-A, all of those are also here too, but they just didn't draw it. I hope I'm making some sense. Now, let's look at it. If there is an ample molecules in the cell called allolactose, we are talking about bacterial cells now. This is all in bacteria. This is all in prokaryotic cells. And you will see in eukaryotic cell is a little bit different. So when this allolactose, the inducer, it's called allolactose, is an inducer, attaches to repressor, then this one, this repressor, cannot attach to promoter. When this is attached to it, when this molecule, disaccharide, this is a disaccharide, it's not a protein molecule, this is a protein molecule, the repressor is a protein molecule, a disaccharide attached to this, it makes it inactive, does not, what does it inactive mean? It means it does not attach to promoter, and when it does not attach to promoter, guess what happens to RNA polymerase? RNA polymerase move along easily and starts doing transcription, and transcription, based on transcription, translation will occur. And these are all protein molecules. Permease, what is that? Trans-acetylase and beta-galactosidase. These are all enzymes. Yes.
Is that how the operon is turned on?
That is how, in this case, that's how operon is turned on by a molecule called allolactose, which is a galactose. Allolactose. Allo, it means different, same as hetero. So it's different than the lactose you learned
Binds
already.
to the repressor.
Bonds to the repressor, very good. Binds to the repressor, then the repressor does not repress anymore. But if there is nothing bound to repressor, then repressor will repress.
That stops it.
Stops it. I hope I'm making some sense. 50 multiple choice in your final. You're looking at them. Your final, there is no essay. 50 multiple choice. I want to pass it through the machine and hand in your final grade. It's on May 25th, right? Am I making some sense? Your final is on Wednesday, May 25th. Let's go. Okay, now inducible enzymes usually function as catabolic pathways. Do you know what is catabolism? What is catabolic pathways? We talked about it in the past. What does that mean? Breaking up. Breaking up. Anabolic, anytime you forget, anabolic is anabolic steroids. People who inject anabolic steroids to build up, they call it anabolic steroids. It means build up. Okay, so that's what happens. Inducible enzymes usually function in catabolic pathway. Their synthesis is induced by chemical signals, and repressible enzymes usually functions in anabolic pathways. Their synthesis is repressed by high levels of end products. Regulation of tryptophan and lac, tryptophan, tryp, and lac operons involves negative control of genes because operons are switched off by the active form of their repressors. Okay, so in this case, in allolactose, and in this case was a tryptophan itself that they repress the inducers. Okay. Positive gene regulation. So let's talk about what is positive gene regulation. You saw two examples of negative regulation, the repressor, and it's not always repressing, it's also making things happen. It's a positive regulation, and I'll show you, we'll talk about it a little bit. Some operons are also subject to positive control throughout. It means turning on the switch, just like the way it is now. Stimulatory proteins such as catabolite activated proteins, which call them CAP, CAP for abbreviation, they call it CAP, CAP. So CAP, for rest of the PowerPoints I'll show you guys, if you know about CAP, the mRNA, where is the ruler? mRNA had a cap on one end and had AAA, or not AAA, poly A on the other end. You remember that? Okay, this is not the same cap. You're talking about different type of cap. This one, you will see it in a minute. The cap is catabolite activator proteins, CAP, an activator of transcription. It means they are activator of transcription, and you will see the pictures. When glucose, a preferred food for E. coli, E. coli is the name of a virus. Name of a bacteria, virus. Name of a bacteria, E. coli is the name of a bacteria, is scarce. There's not much enough glucose in a cell, in a bacterial cell, E. coli. CAP is activated by binding with cyclic AMP. You already know cyclic AMP. Can you refresh my memory? Cyclic AMP was made from what? Do you remember that? ATP. Very good. Cyclic ATP, cyclic AMP, they call it CAMP, was made from ATP, something you know from past. Binding with cyclic AMP, and I'll show you some pictures in here. Activated CAP attaches to the promoter of the LAC operon and increases the affinity of RNA polymerase, thus accelerating transcription, and that is a positive one. When glucose levels increase, CAP detaches from the LAC operon and transcription returns to a normal rate. So this is a positive thing when there is not enough glucose. Okay, anyhow, you will see some pictures and I hope they make sense. CAP helps regulates other operons that encode enzymes used in catabolic pathway, right here. This is an example of positive control. Positive control, it means it's going to happen. The transcription is going to happen. Okay, so you have, this is the case, look at this one. It says lactose present, glucose scarce. Scarce, it means there's not much. Inside of the cell, you do not have glucose molecule. When you do not have glucose molecule, this happens. And what is this? A positive. They call it a positive control. So you have the inactive, this is inactive CAP, and what does CAP stands for? Catabolite activator protein. Okay, so the CAP, the CAMP, cyclic AMP, attaches to inactive CAP, and of course becomes active. When it becomes active, it attaches to the DNA, right here, CAP binding site on DNA, and then of course the RNA polymerase becomes active and this is the operator, moves along and make up those protein molecules on the LAC-Z and LAC-Y and LAC-A. That is in the case of there is scarce amount of glucose in the cell. If there is ample glucose in the cell, lactose present, glucose present, cyclic AMP level is low, little lac mRNA synthesized. So what does that mean? That you have the catabolite inactive CAP, it's in the cell, there is nothing to activate it, okay? So it's just there, and then of course you have allolactose here. This is not attaching to the operator. What happens? The RNA polymerase is not stimulated, does not go through the operon, so there is no protein molecules are being synthesized. Do you remember these protein molecules I gave you? These proteins or molecules are not synthesized. All of these LAC-Z, LAC-Y, LAC-A, they're not synthesized in this case. In this case, when there is not ample glucose molecule, is positive control, it will happen. Okay, this is an example of positive control, and this is an example of a negative control. These are negative control. Okay. So, and here are the bigger picture, and I said cyclic AMP, LAC-M is synthesized. So this will not attach. The RNA polymerase does not attach the gene to the promoter and move along. So no protein is needed because there is enough glucose molecule and lactose in the cell. Differential gene expression, and what is differential? What is differential gene expression? I have not gave you a list of questions, I guess this far in the chapter. I gave you a few of them, and one of you guys returned and took it back, the answer to your homework question. Is that right? How come you retrieved it? Or did I lose it? You handed it, area? Did you hand in your questions, the homework questions? Oh, okay. I thought you put it here and I lost it.
No.
Because I remember in the morning you brought it here, put it here, but anyhow, don't worry about it. Almost all the cells in organisms are genetically identical. Let's stop for a second. Did you know? You already know, but I'd like to emphasize it. That's why I turn it off. The cells in you will see today, the cells in your eye and the cells in your liver, did you know they have same gene? They have same chromosome, they have same DNA.
No.
You did not know that. I think I already talked about it. The cells in your bone are having the same DNA, same chromosome, same genes as the cells in your muscles. Same cells in the skin. From head to toe, I'm just talking about you. I'm not talking about me. Head to toe, all of your cells have the same chromosome, same DNA, same genome. Right? Am I making some sense? How come the cells in the bone look like this, for example, and the cells in the muscle look like this? That's something we're going to discuss. And that's what that sentence means. It means you, and of course, for example, I don't want you confused. If they grab his cells and they want to put them in my body, his cells have these. For example, let me put these. We talked about these at the beginning of the semester. His cells have these molecules, right? They want to put his cells into my cells, and my cells do not have this. My cells have this, right? They're different. So when they put his cells into my cells fight his cells. Is that right? Transplant, rejection, you all know what I'm talking about. That's not what I'm talking about. I'm talking about you have the cells that is within you, within you, the cells, they all have the same gene, the same chromosome, everything is same. I hope I'm making some sense. The cells in your skin cells have X, in my case, have XY. Hairs, they have XX, all the way, from head to toe. XX, XY. That's what that means. Almost all cells in the organisms are genetically identical, except sperm and egg. They're different. That was an exam question last time. What are the differences between meiosis and mitosis? Differences between cell types results from differential gene expression. Differential gene expression. The bone cells make a different type of protein molecule than muscle cells. Liver cells make a different protein molecule than your eye cells. Am I making some sense? Your liver cells make a protein molecule called albumin. Your eye cells make a protein molecule called crystalline, so you can see better. If your eye cells were making albumin, well, you would not be able to see anything. Am I making some sense? And if your liver cells were making, they have the same DNA. They have the same DNA, they have the same gene, they have the same thing. If the liver cells were making crystalline, then osmosis would be imbalanced inside of your body and you would die within 1 day or 2. Okay, so that's what is called differential gene expression. The expression of different genes by cells with the same genome. Same genome. Same genome. Okay, abnormalities in gene expression can lead to diseases including cancer. Okay, so everything comes back to cancer. Gene expression, I bolded for you guys in your PowerPoints that you got it. I will put the most recent, I'm glad you're taking notes, but I will put that after the chapter is revised, I revised my PowerPoints, I will put this, the final version in your, in the docu-share. So you have this one, you know what is important. You know what is important. I bold them for you and hopefully you study them for the exam. Okay, so gene expression is regulated at many stages. What are those stages right here? What are those stages? At one stage, I think this is small for you guys, but this is the big picture, and we will talk about each single, every single one of them. The rest of 10, 20 slides. Okay, so they are signals from environment always is responsible for something. You have signals from environment, whatever that could be, sunlight. You are exposed yourself. I remember when I was in Minnesota and Kentucky, usually females, not much males. Usually they used to go lay out in the sun. Here in California, I don't see that much. You know, people, they lay out in the sun to become tan. Right? Now we have tanning saloons. People go get tan in saloons. But I don't know, that's how Minnesotans or Tennesseans or Kentuckians were like. But, you know, they like to get tan.
They sit out in the sun?
They lay out. No, they go with a bikini.
In the spring and
In the spring, not much in the summer, but early spring, you know, they would go and get tan because they like to be darker like me. And they said, oh, Amir, you have a natural tan. I said, I will not give you any of it. So anyhow, so chromatin is, you all know what chromatin is. Chromatin modification DNA unpacking. So there is a level of controlling genes. Again, don't forget what the chapter is all about. The chapter is about controlling genes. Now we are moving to eukaryotic cells. We are moving into eukaryotic cells. So they are controlling genes at the chromatin level. They are controlling at the transcriptional level, controlling as RNA processing level, controlling as transport to cytoplasm, which I will not talk about this much, but anyhow, degradation of mRNA. As long as mRNA is inside of the cell, translation occur. So if the cell wants to stop translation, that protein molecules to be synthesized, the cell must degrade the mRNA. And then of course translation, and then you have protein processing, and at the end degradation of protein molecules, transport to destinations outside of the cell. Each box indicates a type of regulation. So we will talk about these regulations in here. DNA regulation in blue, that's what this is. RNA regulation in red. RNA regulations in red. And then in orange is protein regulations, these are protein regulations. We'll talk about that in protein regulations in blue. Right here, now you can see it, that the signals from environment can possibly control modification of DNA, and that's DNA gene available for transcription. Of course, this is translation, exon and intron. You know about this very much so. Okay, we already talked, and this is happening in nucleus. Remember, this is the nucleus, and the yellow right here is the cytoplasm of the cell, and of course, this is the cell membrane. Remember that cell membrane, nuclear membrane, nucleus. So these are the events that occurs in the nucleus. And these are the events, the regulations of the gene that occurs in the cytoplasm. Cytoplasm, you have mRNA cytoplasm, degradation of mRNA, and protein processing, and so on and so forth. There is a quick one. Make sure he mentions two terms in here that are
A cell
important.
can get rid of abnormal or damaged proteins and limit the lifetime of functional proteins by means of selective degradation. The cell marks the protein for destruction by attaching small ubiquitin proteins. Giant protein complexes called proteasomes recognize ubiquitin and break down the tag protein.
Sweet and short. Two terms he threw at you that you should know. Well, it will come up. Proteasomes, they're huge protein molecules that they degrade old protein molecules. They break them down. They're like incinerators of the cell. And then, of course, those old protein molecules have to be labeled. If they are not labeled, then they will not be destroyed by ubiquitin, which he mentioned. Okay, you will see, you will encounter them, and I bold them and enlarge them for you in the PowerPoint. Protein processing. Another video.
Often, polypeptides must be modified in some way to produce functional proteins. For example, a polypeptide is cleaved in 2 or more polypeptide chains. Other molecules might be attached to the protein, or the protein might be phosphorylated. Some proteins need to be transported to specific destinations.
Right. You already know about most of these things. The first one you didn't know.
Translation of specific mRNAs may also be blocked by regulatory proteins that bind to sequences within the untranslated region at the 5' end of the mRNA molecule, modulating the amount of protein produced.
This is ribosome. The large subunit, small subunit of ribosomes, they cannot come together because of this regulatory protein binding. If they do not come together, then translation will not occur. Transcription already occurred, but translation will not occur. Okay, regulations of chromatin structures. The structural organization of chromatin helps regulate gene expression in several ways. Okay, so gene within highly packed heterochromatin are usually not expressed. Okay, I talked about this, the heterochromatin, chromatin, when the chromatin, this is chromosomes, when they are packed together, when the chromatin, the chromosomes material are packed together, there is no transcription or translation. The eochromatin, the chromosomes that are loosely packed, we call them eo. Eo, it means true. Eochromatin, then they can easily unfold and transcription occurs. Am I making some sense a little bit? So heterochromatin, they are chromatin that they are packed together. And that is hard to transcribe. Transcription occurs. And right here. Structural organization of chromatin helps regulate gene expression in several ways. Genes within highly packed heterochromatins are usually not expressed. They usually do not get transcribed. Okay. Chemical modifications to histone. Can somebody refresh me or refresh my memory? What is it? Histone. We already talked about it several times. The protein molecules that DNA wraps around it. I have a protein molecule here inside of my chromosomes, a protein molecule here, and DNA right here, DNA wraps around it. Goes to the next one. DNA. These protein molecules, my fingers, like my hand, is histone, and the blue cord is DNA. You have them. That's what your chromosomes are made up of, protein and DNA. Okay, and then they are, when they are not multiplying stages, then they're called chromatin. Remember that those terminologies I gave you somewhere along the line? Okay, the structural organization of chromatin helps regulate genes within highly packed chemical modifications of histones. So you will see that here in a minute. There are 2-3 ways to modify histones. I will talk about it. And DNA chromatin influence both chromatin structures and gene expressions. Let's talk about it. Okay, I'm talking about histones. Histones become acetylation. It means you add an acetyl group. At the beginning of semester, first exam, I gave you a list of functional group: acid, amino, acetyl group, and I did list them for you here. That's the acetyl group. It means this attached to another carbon or so. So this one is acetyl group. Acetyl groups are attached to positively charged lysine. Lysine is what? Does anybody refresh my memory? What is a lysine? 20 what? 20 amino acids. And lysine is one of those 20 amino acids. So what happens is acetyl group attaches to lysine right here in histone tails, and this loosen up chromatin. So that compact chromatin that I talked about right here needs to loosen up. How does it loosen up? By adding an acetyl group to it, so it will loosen up. Okay. Acetylation promotes the initiation of transcription. The addition of methyl group right here, methylation, can condense chromatin. So when you add methyl, this is another functional group I gave you at the beginning of semester. I was hoping you'd know them for rest of semester. So when you add methyl group, CH4, it will what it will do to the chromatin. It will condense it. So acetyl group will loosen it up, and methyl group will condense it. Okay. So when it is condensed, then no expression. When it is loosened up, there is expression of the genes. The addition of phosphate group, another one, phosphorylation next to a methylated amino acid can loosen up chromatin. That's another one. So phosphorylation can loosen up. Here they are, a few examples. Here is your histone. These are the tails of the histone tails right here. DNA, double helix, wraps around, as I showed it to you, the cord. I talked about this in the past, but refresh your memory. These are protein molecules, and at the end, these are amino acids, amino acids, amino acids, amino acids. So what happens in this case, histone tails protrude outwards from the nucleosomes. Unacetylated, does not have acetyl group, the green dots. They are acetyl group attached to the tail of the histone, and what will acetyl group? It will loosen up chromosomes. The chromatin, it loosen it up. And what is the advantage of loosening up chromatin? Anybody? What is the advantage of loosening up chromatin? Well, it will do, yes.
Then transcription can occur?
Then transcription will occur. When transcription will occur, then the protein molecules is synthesized. That's the advantage of it. Okay? So you're kind of turning on genes. in here. You're turning on genes. And this one right here, both of them, this one is turning off genes by methylation. Okay, I hope I'm making some sense. Okay, so these are all examples of, these are chromatin modifications. Chromatin modifications. Either chromatin modifications, either loosening it up, condensing it, or loosening it up. I hope I'm making some sense. I hope you're sticking with me. Okay, epigenetics, I do have a video for you guys to watch, but you know, so far I know I gave you the impression, that either it is your genes that make you up or the environment, two separate concepts, the environment and nature or nurture. Have you heard of that? Nature or nurture, the environment. Okay, you've heard of that in psychology classes, in, I don't know, biology classes, your other classes, nature versus nurture. But what epigenetics is, it's a combination of both. It's both that are together. And we are understanding that a little bit more and more and more now. Although the chromatin modification just discussed do not alter DNA sequence, they may be passed to the future generations of the cells. So that is the genetics, okay, the nature part. The inheritance of traits transmitted by mechanisms not directly involving the nucleotide sequence is called epigenetic inheritance. Examples, identical twins, one is normal and one is with schizophrenia. It's a 10-minute video. I got it from YouTube because I thought, how can I make this point more clear? And this guy, bless his heart, he did a great job of going. Again, you will get this. You can show it to your family members if you guys are. I hope now it's going to take half an hour to. Oh, we did it.